So I think one of the first things about this vaccine is that we've developed this and really aggregated technology across the country. We've got a number of collaborators working in different areas of expertise, as well as our contract manufacturing, who is responsible for making the vaccine. And so it's with these world-class researchers and partners that we've been able to put together this innovative product called Berger. It all starts at Iowa State University. Iowa State University has the ZMax cells. Dr. Zhang will be talking later on about some of his work that he's done with the ZMax cells in isolating PRRS viruses from the field. Those isolates are then sent down to our contract manufacturing organization, and they propagate that virus up in order to make enough of the antigen for the actual vaccine. The vaccine is then assembled by combining with an adjuvant. It's a novel proprietary adjuvant that we use in this formulation, along with a nanoparticle that actually is adhering the virus to it and then presenting the virus later on to the antigen-presenting cells. And of course, this vaccine is delivered intranasally into the pig's nostrils at the time of administration. The ZMax cells are particularly sensitive to PRRS viruses. The macrophage alveolar cell was invented by Dr. Zuckerman, who is sitting here today. And this cell line has, we have not found a PRRS that it doesn't grow. And so we're very excited to be able to get the isolates from the field and very quickly grow that isolate up into acceptable virus concentrations for vaccine production. We've taken the process up to 200 liters. There's no reason why we couldn't go higher. And these are just some pictures here of traditional kind of antigen production. So we're using fairly standard techniques for growing the virus. It is then, importantly, inactivated. And we've taken great care with this. This is a validated process for virus inactivation. We confirmed that the virus is, with the BEI concentration that we're using, is inactivated within 12 hours. However, we do extend that to a 24-hour period to just further ensure complete inactivation. And we also then, at the end of inactivation, we test this antigen by passaging it through ZMax cells and looking for the ability to kill those cells, and we passage that 3 times to verify that there's no residual virus that has not been inactivated. We then go through some purification processes. The interaction of the virus with the nanoparticle is based on hydrophobic as well as electrostatic interactions, and we don't want to have extraneous proteins in this formulation, which could potentially bind up those very important sites. So the process that we put in place and that we've been working with for the last year now has the ISU sample comes in and goes to the contract manufacturing after the screening that is passaged up 2 times. These are seed banks that are put down in a freezer, and then those are used to further prepare the antigen. We've got cell cultures that are continuously being maintained at contract manufacturing sites for both the flu and for the PRRS, and so that they can get into antigen production quickly. And we look at somewhere between 8 weeks and 11 weeks, 8 weeks being closer for the first time that isolate has been used in an autogenous formulation, 11 weeks if it's the second or subsequent runs after that. So I'll talk a little bit about the adjuvant. What is an adjuvant? And it's really, it means to help. And adjuvants have been identified in vaccine formulations to help moderate and stimulate the appropriate immune responses. So our adjuvant is combined with these nanoparticles and stimulates a strong cell-mediated type of response. And you'll hear me talk about whole cell lysate. This is actually a bacterial whole cell lysate. And many bacteria, many organisms themselves have adjuvant properties. And in fact, some vaccines are based exclusively on antigens that themselves behave as an adjuvant. It's a mycobacterial whole cell lysate. If you're familiar with Freund's adjuvant, it's a very good and powerful adjuvant. It's also quite reactive. This is not based on tuberculosis. It's another mycobacterial species. And it's a process where we grow the bacteria, then we purify that bacteria, we lyse it, and eventually we get a whole cell lysate which contains a number of different kind of immune stimulating factors in it. We do something I think is unique with an adjuvant, and that is because of the effects of this adjuvant, we can take it on our ZMAX cells and actually stimulate those ZMAX cells to produce TNF-alpha. And we quantify that. With each batch, then, we can dial in not just the concentration of the adjuvant itself, but we can also get some perspective of the potency, and we can see slight variation from batch to batch. That allows us to optimize that formulation when we're putting it together with the antigen at the end. The nanoparticle is, and sometimes we refer to this as the SATX or surface array technology, and as I said, this particular formulation has been optimized for viruses to make sure that the virus adheres electrostatically, hydrophobically with the actual nanoparticle. And this nanoparticle then acts as sort of a delivery device, if you will, to the antigen-presenting cells, the macrophages, or the dendritic cells that are going to take up these nanoparticles that are decorated with the PERS antigen and the adjuvant that is combined in this vaccine and acts as a very powerful immunostimulant. These are just some of the quality control aspects of our nanoparticles, making sure that the charge is right and making sure that the size distribution is right, so it's not too large for uptake by these antigen-presenting cells. And so at the end of the day, our manufacturing process is quite compatible with what you find in traditional vaccine manufacturing facilities. And I've been working in vaccine manufacturing for over 35 years, and so it's a very simple, easy formulation, mainly because we bring that technology into the contract manufacturing organization, and what they're doing is simply assembling these components together in the right way to make the final vaccine. It's low cost, very scalable. It doesn't require the use of like freeze drying or lots of solvents in order to extract different kinds of proteins and things like that. And we found it to be very easy to assemble and something that we can train other people how to use. And this is sort of a diagram of really conceptually what is happening when these three components are brought together into the barricade formulation. And that's our label. This is the label that would go on to a product that would be delivered. So USDA approved, this is the actual approved label.